Welcome back, everyone. Continuing through our look at the blood, we are now starting to look at some of the red, white blood cells and some of the other components that are in the blood aside from the plasma. So we're going to spend 2 lectures talking a little bit about red blood cells. Then we will talk about white blood cells and the immune system, and wrap up with platelets. So let's go ahead and start with our not really attendance questions for the day. First, what level of protein structure is it when 2 or more proteins stick and work together as a single unit? And next, what organelle uses oxygen and glucose to produce ATP? Go ahead and try to answer those. Hopefully you've done that. So let's take a look to see what the answers are. What level of protein structure is it when 2 or more proteins stick together, work together as a single unit? Well, remember, primary was when we had a string of amino acids. And then when those amino acids started interacting with each other and with the environment, they started to either coil up or fold up or take on these random twists, and we called that level secondary. And then when those coils and folds and twists started interacting, it folded up and took on a 3-dimensional shape, and that was the tertiary structure of a protein. And that tertiary structure, that is when we started calling it a protein. But some proteins actually need to stick to other proteins in order to do their intended job. And when that happens, when we have 2 or more proteins stuck together acting as a single unit, we say that is a protein with quaternary structure. Next, what organelle uses oxygen and glucose to produce ATP? That is the mitochondria. So both of these will play a role in today's lecture. If you don't remember those things, go back and look at Chapter 2 for the first question and Chapter 3 for the second question. Now, in the previous lecture, we talked about protein, the extracellular matrix of blood, the liquid part of blood, all of that white background in this image. But there's a lot of other stuff going on in this microscope. So what we see here are lots and lots of these. I called them little pink donuts or Cheerios. Those are the red blood cells. And then these larger things with the weird purple blobs inside, those are the white blood cells. And these little purple flecks, or sometimes they don't stain the same, and sometimes they're little pink flecks, those are platelets. So red blood cells, white blood cells, and platelets, these are called formed elements. The formed elements are the parts of blood that are cellular in origin. Notice I did not say they are cells. They are cellular in origin. And as we talk about each of these cell types over the rest of this chapter, we will see what their functions are, what their origins are, and why I did not exactly call them cells. So the formed elements, they all have something in common, and that is that they are all cellular in origin. They come from cells, or they are cells. Some are cells, some are not. So speaking about that, some are not, let's talk about red blood cells and platelets. Neither of them are true cells. We're going to see today that red blood cells, they start out as a typical cell, but they go through some changes during their development, and the result is they do not have any organelles once they finish developing. They don't have a nucleus, they don't have the other organelles So we do not classify them as a cell. They don't have all of the features needed to be considered a cell. Platelets take that even further. Platelets start out as a gigantic cell, and pieces of it flake off, and that's what platelets are. They're flecks of a bigger cell. So they're all cellular in origin, but red blood cells and platelets are not cells themselves. Now, mostly they are amitotic. So what does that mean? Well, amitotic means without, A means without, and mitotic refers to mitosis. They don't go through mitosis, generally. So what does that mean? Well, red blood cells, they can't go through mitosis. They don't have a nucleus. And platelets can't go through mitosis. They're not even cells. They don't have anything. They are just pieces of a cell. And white blood cells, well, we're going to spend a lot of time with those soon, and they are part of our immune system. They can go through mitosis. They tend to go through mitosis when there's some sort of infection, but they're not undergoing regular mitosis. They all come from the red bone marrow. Every part of blood comes from the red bone marrow. So now we've gotten the formed element stuff out of the way. Let's talk specifically about red blood cells. And now that we are moving into some more detail, we're going to stop using that word or those words. We're going to use the proper name, and that is erythrocyte. So erythro means red, cyte means cell. So red cells or red blood cells. And that's what we see here. Now, they're kind of round. We say they are discoid shaped. They're kind of round, but imagine you have a round piece of clay or Play-Doh, and you put one finger on top and your thumb on the bottom, and you squeeze a little bit. So this is from the side. We can see that there's a dimple in the top and a dimple in the bottom. Here is that dimple from above. So we say they are biconcave. Biconcave, well, concave means sunken in, curved inward. Biconcave means it's happening twice, one on each side of the cell. And this creates a very high surface area to volume ratio. That means for as much as it can hold, the volume, the area on its surface is very, very big. High surface area compared to the volume. Like I said earlier, it does not have any organelles, and that's why it's got this caved-in appearance. It starts out round and plump like a traditional cell, but as everything is kicked out, all the organs are kicked out, it caves in on itself. Now, everything in our body is mostly water, and that's true for red blood cells. But if we ignore the fact that it's mostly water and we just look at the non-water part of the cell, well, other than water, the cell is 97% hemoglobin. Erythrocytes are packed full of a protein called hemoglobin. Hemoglobin is the protein that is what carries oxygen. When oxygen travels in our blood, almost all of that oxygen in our blood is bound to hemoglobin. So this cell is packed with hemoglobin. Now, think about those things. High surface area to volume ratio. It's biconcave. It's got no organelles. It's mostly hemoglobin if we don't count the water. Let's use that as a thinking cap question. Why are those qualities important for erythrocytes? And as a hint, what's the job of an erythrocyte? So try to answer that before you listen to what the response is. Well, what's the job of an erythrocyte? Well, to carry oxygen. Why are these things important for that? Well, the high surface area to volume ratio, because it is biconcave. That means it's got a lot of surface. Why would we want a cell with a lot of surface? The more surface area there is, the more space there is for oxygen to move in and out of the erythrocyte across the plasma membrane. This makes it really, really efficient for loading and unloading oxygen. Other than water, it's 97% hemoglobin. Okay, so hemoglobin, we just said, is the molecule that carries most of the oxygen. So pack it full of hemoglobin and we're great. All that oxygen getting in binds to hemoglobin. This guy is prepared to deliver some oxygen. But the kind of confusing one is this organelles. It doesn't have any organelles. How does that help this guy carry a lot of oxygen? Well, organelles do things. And to do things, a lot of times that requires ATP. Who makes ATP? The mitochondria. So the mitochondria is not there to make ATP, and the organelles are not there to do things that require ATP. Why is that a good thing? Well, where does that ATP come from? From glucose and oxygen. We don't want the cell that's delivering oxygen to be using the oxygen while it's doing that. Get rid of its ability to use oxygen. Get rid of its ability to do anything except carry oxygen. Now it's a really, really efficient oxygen delivery unit. So let's look a little more at hemoglobin. The function of an erythrocyte, we've said now a few times, is to deliver oxygen. It's an oxygen transport. This is what hemoglobin looks like. It's got a quaternary structure. So it's 2 or more proteins stuck together. Well, really, hemoglobin is 4 proteins stuck together. We can see we've color-coded it here. There's this dark brown, and there's 2 dark brown areas called the alpha domains. There's 2 alpha domains, and there's 2 of these light brown areas, beta domains. So we've got an alpha domain, a beta domain, another alpha domain, and another beta domain. So there are 4 proteins working together. Now, right in the middle of each one is something called a heme pigment. That's what we see right here. Both of the alphas have a heme pigment. Both of the betas have a heme pigment. Let's lift that heme pigment out and take a look. Here is our heme pigment. One molecule of hemoglobin has 4 heme pigments, 1 in each of those subunits. Here is the heme pigment, and right in the middle of it, what do we see? We see a single iron atom. Iron really likes oxygen. When you see something rusting, most of the time that is iron with oxygen attached to it. So, one iron atom can attach to 1 oxygen molecule. Now, think on that. How many oxygens can 1 hemoglobin molecule carry? How many oxygens can 1 hemoglobin molecule carry? 4. They don't always have 4, but when they do, they are as packed full as they can get. 1 oxygen molecule attached to each heme pigment. Now, each red blood cell, each erythrocyte, has lots and lots and lots of hemoglobins. Each one can carry 4 oxygens. So one erythrocyte can really, really pack in a lot of oxygen. But in addition to carrying oxygen, hemoglobin is a buffer. What is a buffer? Think back to chapter 2. A buffer is anything that resists pH change. Anything that resists pH change. Both of these things, the fact that it carries 4 oxygens per hemoglobin, and the fact that hemoglobin is a buffer, those two things will come back when we get to the respiratory chapter and when we get to the pH regulation chapter. So make sure to make some notes of that. Now let's start to see, we've learned what do erythrocytes do. Let's see how are erythrocytes produced. And to do that, we're going to have to learn some terminology. One term is hematopoiesis. We're going to see the word poiesis a lot this semester. Poiesis means formation of. Hemata means blood. Hematopoiesis is the formation of whole blood. Hematopoiesis takes place at the red bone marrow. So red bone marrow makes blood. Red bone marrow performs hematopoiesis. But there are different parts of hematopoiesis, just like there are different parts of the blood. So when we are making red blood cells, erythrocytes, that process is called erythropoiesis. Erythropoiesis is the formation of erythrocytes. When we make platelets and when we make white blood cells, it's another part of hematopoiesis with different names. So hematopoiesis, making blood. Erythropoiesis, making red blood cells. And it doesn't matter which cell type we are making. We begin with a hematopoietic stem cell. A hematopoietic stem cell. Hematopoietic stem cells are stem cells that are constantly going through mitosis to create other cells. We saw different types of stem cells back in Bio 137. These behave just the same, but these cells make blood cells. So another name you might hear them called is hemocytoblast. Hemocytoblast. Hemo for blood, cyto for cell, blast to give rise to. Now, as we're making the erythrocytes, these hematopoietic stem cells are going to produce a cell that's going to go through some changes and eventually eject its organelles, since we're making erythrocytes. So here is the step-by-step production of an erythrocyte. We begin with that hematopoietic stem cell, the hemocytoblast. If it's going to produce an erythrocyte, it goes through cell division. One of those cells remains a hemocytoblast. The other cell becomes something called a pro-erythroblast. So the hemocytoblast goes through mitosis. 2 new cells. One of those cells is still a hemocytoblast. It replaces itself. The other cell is going to continue on to make an erythrocyte. So that cell that's going to continue on is called a pro-erythroblast. It goes through some changes. Ribosomes are going to start being produced. Ribosomes are going to start making lots of proteins. Some of those proteins, a lot of those proteins, they're going to be what? They are going to be all of that hemoglobin that we just saw. This is called a basophilic erythroblast. we go through some more changes. Polychromatic erythroblast. We're making more and more and more hemoglobin. Next, we become an orthochromatic erythroblast, making even more hemoglobin until eventually we're packed pretty full of hemoglobin. We kick out the nucleus and the other organelles. We become something called a reticulocyte. And now we exit the bone marrow. Sometimes the reticulocyte exits the bone marrow, but definitely by the time we get to full developed erythrocyte, we have left the bone marrow and are circulating in the blood. It is not uncommon to find some reticulocytes circulating in the blood. But erythrocytes, that is the final product. So how do we control production? How do we know when we need to make red blood cells or when we don't? Well, think back to the end of the endocrine system. We talked about erythropoietin, EPO, made by the kidneys. When there is low oxygen detected by the kidneys, that stimulates the red bone marrow. Well, we stimulate erythropoietin, which stimulates the red bone marrow to make more erythrocytes, which increases the amount of hemoglobin in the blood. Well, low red blood cells can also stimulate erythropoietin. Low hemoglobin levels can stimulate erythropoietin. So all of these things are kind of related. Low oxygen levels, low red blood cells, and low hemoglobin, they all kind of go hand in hand. And each of them will stimulate erythropoietin. Testosterone can also stimulate erythropoietin, which is why males tend to have a higher hematocrit. Males tend to have higher testosterone. Testosterone stimulates erythropoietin, so males tend to have a higher hematocrit. Remember, hematocrit is that packed red blood cells when we do that centrifuge. So now up here I've got something about blood doping. This is something that comes up commonly during bicycle races. Sometimes during the Olympics, you start to hear about blood doping scandals. Blood doping is what happens when you inject synthetic erythropoietin. Erythropoietin, if you inject it, what's going to happen? You're going to make more red blood cells. Why would you want to do that? Well, more red blood cells, you can carry more oxygen, which gives you a performance advantage in sporting events. It's hard to detect because you naturally make erythropoietin, so seeing erythropoietin in the blood would not look abnormal at first. But we run a risk with this. Think about why that might be. If you're blood doping and you're making more red blood cells, your hematocrit goes up. If your hematocrit goes up, what happens? Your blood gets thicker. It gets more viscous. Remember that word? Viscosity. So if blood is more viscous, if it has higher viscosity, it's going to have more resistance to flow. Connect that back. It's going to be harder for that blood to move through your blood vessels, so it makes your heart work harder. Your blood pressure increases. Blood doping to increase your hematocrit, yeah, it helps you carry more oxygen, but it can cause a lot of problems up to and including death because it makes your heart work so hard. So B12, vitamin B12, and folic acid, these are both needed for red blood cell formation. So the B12 and the folic acid, one, they're both needed for DNA production. They are also both needed to absorb iron from your diet, if you don't have iron in your diet being absorbed, and if you don't have B12 and folic acid, well, you can't make adequate hemoglobin and red blood cells. We're going to come back to that in just a moment. But here is our homeostatic feedback loop. We have normal levels of oxygen, but when we have low oxygen, our kidneys detect that and stimulate the red bone marrow to make more red blood cells by using erythropoietin as a stimulant. We make more red blood cells, erythropoiesis. Our red blood cell count goes up. Now we can carry more oxygen, so now we have more oxygen carrying capacity, and that cuts off. Once we make these red blood cells, how long do they live? Well, not very long. Cellular standards. Some cells, live our whole life or close to it. Red blood cells, erythrocytes, only live about 120 days. That's not very long for a cell. Why is that? Well, it's kind of rough traveling through the bloodstream. They're constantly bumping up against the walls of the blood vessels, and it causes them to, have pieces of them flaked away, lots of damage occurs. But since there's no internal cellular machinery, they've kicked out all of their organelles, well, they can't repair that damage. So after about 120 days, they're not very useful. And that's not bad because we're constantly making more red blood cells, so losing them constantly is okay. At the end of that about 120 days, macrophages, well, macro means big and phage means to eat. These are big cells that eat debris. So they are destroyed by macrophages, and those macrophages, they're all over our body, but most of the destruction of an erythrocyte happens in the spleen. It also can happen in the liver, and it can happen in the red bone marrow. And when we break a red blood cell down, since it is mostly hemoglobin, aside from water, what happens to that hemoglobin? Well, it's not just going to be pure waste. It's got a lot of useful stuff in it. So we have our hemoglobin, and when the red blood cell is destroyed, that hemoglobin is salvaged, and it's split into its two components. It's split into the protein portion, called globin, and that iron-containing heme pigment. Let's first look at the globin. What happens to the globin? Well, it's a protein, so what's going to happen? It's going to be broken down into its component amino acids. All proteins are made of amino acids, and those amino acids move back into the bloodstream, redistributed throughout the body, and used to make more proteins. Maybe more globin, maybe some other protein. Whatever we need, these amino acids can be used to build those proteins. But the other part, the heme, is broken down. It's broken down into the iron, and the other portion is called bilirubin. So let's follow the iron part of the heme. Iron is attached to something, and we store it as a molecule called ferritin or hemosiderin. But we're just going to talk about the ferritin. Iron stored as ferritin in the liver, and then the liver attaches that iron to a carrier protein called transferrin. Transferrin is one of those proteins that we talked about that helps transport iron in the blood. Now, that iron will eventually make its way to the red bone marrow, and we will just make some more hemoglobin out of it. Back up here to the heme, Part of it was iron. We just saw what happens to that. The other part is bilirubin, and bilirubin is also moved into the liver. The liver takes the bilirubin and it's going to attach it to some substances in the bile. And then the bile is secreted into the small intestines. Bacteria start to break some of it down. And when it breaks that down, well, interesting fact, that's why feces is brown. When we break down bilirubin, it causes brown to be placed into the small intestines, and that brown is what stains the feces brown. That's why one of the common questions at the doctor is about stool. They're going to ask, what does it look like? They're not just being creepy. They want to know things like, what color is it? Because if it's something like white or light brown clay color, something like that, that can indicate a problem with the liver. So, as that moves through, most of that bilirubin exits the body in the feces. Some of it moves back into the blood, along with some of that iron and everything. It is going to be recycled. So let's see an animation. I don't know if it's going to have sound. I'm going to assume that it does, but this is going to show us what it is that happens to those red blood cells. Maybe, if I can get it to play.
When old red blood cells rupture, the released hemoglobin is ingested by macrophages. The globin chains of hemoglobin are broken down to individual amino acids that are metabolized or used to build new proteins. Iron is released from the heme of hemoglobin. The remaining structure is converted to biliverdin, which is then converted into bilirubin. Iron is transported by transferrin in the blood to various tissues for storage, or to red bone marrow for making new hemoglobin. Free bilirubin is transported by albumin in the blood to the liver. Liver cells make conjugated bilirubin, which is excreted as part of the bile into the small intestine. Intestinal bacteria convert bilirubin into bilirubin derivatives, which contribute to the color of feces. Some of the bilirubin derivatives are absorbed into the blood and excreted from the kidneys in the urine.
All right. So now let's talk about some things that can go wrong with our erythrocytes. Well, one thing is anemia. And anemia is not a disorder. It's actually a class of disorder. There's several different types of anemia. One of them is called hemorrhagic anemia. Well, first let's talk about what anemia actually is, what the different things that are called anemias have in common. Well, anemia is just an inability to adequately carry oxygen. In hemorrhagic anemia, the reason we are not adequately carrying oxygen is because we're bleeding and losing blood volume. Hemorrhage is just bleeding. Hemorrhagic anemia is you can't carry oxygen well because you don't have enough blood. Next, iron deficiency anemia. Here, you do not have adequate iron. Maybe it's a dietary thing. Maybe you're not eating enough iron. Maybe your body is not handling iron well. For whatever reason, you don't have enough iron, so you can't make hemoglobin well, and therefore you can't carry oxygen. In pernicious anemia is, there's a couple of different causes. One is autoimmune. Your immune system attacks the lining of your stomach. Well, your stomach makes something called intrinsic factor. Now we're going back to what I talked about a little bit ago about B12 and folic acid. So if you have this autoimmune disease attacking your stomach lining, well, your stomach lining makes intrinsic factor. Intrinsic factor is what is needed for your body to absorb B12, which is tied into iron handling and, like we said, DNA synthesis. So low iron, low B12, low intrinsic factor, all of that together, you can't make hemoglobin. And I have something here interesting where I say weight loss surgery tie-in. With the rise of weight loss surgery, there was a rise in pernicious anemia, but it wasn't because of autoimmune diseases. Well, some types of weight loss surgery remove a part of the stomach, and with that comes the stomach lining. So if you are removing a significant amount of stomach lining, you're going to have less intrinsic factor, less B12, and you can't make hemoglobin well. That's also a type of pernicious anemia. Next, renal anemia. Here, the kidneys are either not detecting the low oxygen or not releasing erythropoietin. Whatever it is, the kidneys are not telling the red bone marrow to make erythrocytes, and therefore we can't carry oxygen as well. Next, aplastic anemia. In this case, the red bone marrow is destroyed. This could be because of radiation for certain types of cancer, so it could be intentional, short-lived. It could be because of an autoimmune disease. It could be because of a number of things, but the result is no red bone marrow. You can't make red blood cells. So that's aplastic anemia. Hemolytic anemia. In this, the red blood cells themselves are being destroyed. Next, thalassemia and sickle cell anemia. These are similar. These are both genetic. They are both disorders that result in the hemoglobin molecules being misshapen. In sickle cell anemia, that misshapes the entire red blood cell so that it's kind of hook-shaped or sickle-shaped. It tends to be more prominent in Africans. Thalassemia results in the red blood cells kind of having a weird bullseye appearance, and that tends to be more common in people of Mediterranean descent. But the result in both of these is the same. The red blood cells cannot adequately carry oxygen, again making these anemias. Now at the other end, there's something called polycythemia. Poly means many. So polycythemia is what happens when you have too many red blood cells. And we've talked about back in blood doping, if you have too many red blood cells and your hematocrit goes up, well, that can be problematic. In polycythemia, this is not always, but it's often the result of a cancer either in the red bone marrow or in one of those cells on the way to becoming an erythrocyte, so that we have too many erythrocytes being produced, hematocrit goes up, blood becomes harder to circulate. All right, so that is the end of the erythrocyte lecture. Next, we're going to be talking about blood groups, which really ties into erythrocytes. So it's really erythrocytes part 2. And I will talk to you in that lecture.